Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore two of the most fascinating and crucial concepts in the field, penetrance and expressivity. You might think that inheriting a specific gene means you will definitely show the associated physical trait. However, real-world genetics is rarely that simple. Sometimes genes hide, and sometimes they show up in unexpected ways. Let us dive in and discover why. To start our journey, let us imagine you and your friends are all using the exact same recipe to bake a chocolate cake. You would expect every single cake to come out looking exactly the same, right? In genetics, this recipe is your DNA, specifically your genotype. The final baked cake is your physical trait, or phenotype. But what happens if a friend follows the recipe perfectly, yet their oven malfunctions? The cake simply does not bake at all. Similarly, a person can have the exact genetic recipe for a trait, but for some reason, that trait never actually appears in their body. This surprising phenomenon brings us to our first major concept. Penetrance is essentially a binary, yes or no question. Does the individual show the trait, or do they not? When every single person who carries a dominant mutant allele actually shows the mutant trait, we call this complete penetrance. However, if some individuals carry the allele but look completely normal, we are dealing with incomplete penetrance. Having the gene does not guarantee showing the trait. This challenges the simple Mendelian idea that a dominant allele always results in a dominant phenotype, showing us that genetics is influenced by complex internal and external factors. Now that we understand what penetrance is, let us look at why it matters so much in the real world. Understanding penetrance is absolutely critical in medical genetics and genetic counseling. Imagine a patient tests positive for a genetic mutation linked to a severe disease like breast cancer. They will immediately want to know if they are definitely going to get sick. Because of incomplete penetrance, a doctor can explain that having the gene only gives them a certain percentage chance of developing the condition, not a guaranteed fate. This knowledge helps patients make informed decisions about preventative care without causing unnecessary panic. Since we know that not everyone with a gene shows the trait, we need a way to measure this mathematically. This formula calculates the penetrance percentage, which tells us how often the gene actually does its job. The variable p represents this penetrance percentage. The symbol Ne stands for the number of individuals who actually express the phenotype, meaning they show the physical trait. The symbol Nt represents the total number of individuals who possess the specific genotype in question. By dividing Ne by Nt and multiplying by 100, we convert this ratio into a clear percentage. If the value of Ne increases while Nt stays the same, the penetrance goes up, meaning the gene is more consistently expressing itself. Before we move on, let us see this formula in action with a concrete example. Let us look at a real-world genetic condition called polydactyly, which results in extra fingers or toes. Suppose a geneticist finds that 40 people carry the dominant allele for polydactyly. This is our total, Nt. However, only 32 of these individuals actually have extra digits. This is our expressed number, Ne. We plug these numbers into our formula, dividing 32 by 40, which gives us 0.80. Multiplying by 100 gives us a final answer of 80%. Physically, this tells us that if a person inherits this specific dominant allele, they have an 80% chance of actually developing extra fingers or toes. So, we have established whether a trait appears or not. This brings us to our second major concept, expressivity. Let us go back to our cake baking analogy. This time, everyone's oven works, and every single person successfully bakes a cake. The penetrance is 100%. However, when you look at the cakes, some are slightly brown, some are perfectly dark chocolate, and some are almost burnt black. The intensity of the chocolate color varies wildly. In genetics, this variation in how strongly a trait shows up is called expressivity. While penetrance asks if the trait appeared at all, expressivity asks how severe or noticeable it is once it is there. Now that we understand the definition, let us explore why variable expressivity is incredibly important for understanding genetic diseases. When a genetic trait shows variable expressivity, it means that two people with the exact same mutant allele might have vastly different physical symptoms. One person might have a very mild, almost unnoticeable version of the trait, while another person might suffer from a severe, life-altering version. For instance, in Marfan syndrome, one patient might only be unusually tall, while another might have life-threatening heart defects. Recognizing this concept helps medical professionals look for a wide range of symptoms within a single family, ensuring accurate diagnosis and proper care. To quantify this sliding scale of severity, we use a formula to calculate an average severity score. The symbol S average represents this overall picture of how strongly the gene expresses itself in a population. The numerator is the sum of all individual severity scores, where each person showing the trait is given a score based on how intense their symptoms are. The denominator, NE, represents the total number of individuals who actually express the trait, just like in our penetrance formula. By dividing the total sum of severity scores by the number of affected people, we get a standard mathematical mean. If the individual scores increase, the average expressivity will also increase, indicating a generally more severe manifestation. Let us apply this severity formula to a classic example of variable expressivity, the coat color of beagle dogs. Suppose a breeder has a litter of 5 puppies that all share the exact same genotype for a specific spotting gene. The penetrance is 100%, so our NE is 5. The breeder grades the white spotting on a scale from 1 to 10. The puppies receive scores of 2, 4, 5, 8, and 9. We add these individual scores together to get a total sum of 28. We then divide 28 by the 5 puppies. The final average expressivity score is 5.6 out of 10. This tells the breeder that this specific genetic line tends to produce a moderate amount of spotting on average. Now that we have calculated both concepts, let us look at a visual comparison to cement our understanding. On the left side of this diagram, we see incomplete penetrance. Notice how some circles are fully colored purple, while others remain completely white. The trait is either entirely present or entirely absent, even though all these individuals share the same genotype. On the right side, we see variable expressivity. Here, every single circle has color, meaning penetrance is complete. However, the circles range from light lavender to deep dark purple. The trait is present in everyone, but the intensity varies wildly. This perfectly illustrates the difference between whether a trait appears and how strongly it appears. This visual comparison leads us to a very helpful analogy for remembering these two concepts. Think of penetrance as a simple on and off switch on your wall. It is a quantitative measure that determines if the light bulb turns on at all for a specific genotype. Expressivity, on the other hand, acts like a dimmer switch. Once the light is on, the dimmer switch controls the qualitative intensity, or brightness of that light. Together, this on and off switch and dimmer switch explain why real-world genetics is rarely as simple as Mendel's basic pea plant experiments. Genes constantly interact with complex environments and backgrounds to shape living organisms. To wrap up our lecture, let us review the most important ideas we have covered today. First, penetrance is a quantitative measure of whether a trait is expressed at all. Remember that incomplete penetrance means having a mutant gene does not guarantee you will show the trait. Second, expressivity is a qualitative measure of how much the trait is expressed once it is present. Variable expressivity explains why the exact same genetic mutation can cause mild symptoms in one person and severe symptoms in another. You have now mastered the fundamentals of how genes express themselves in populations. Great job!